Hi friends, welcome back to our channel. Today's topic is yeast to hybrid system. It is a molecular biology technique. Why it is performed? It is performed to detect the protein-protein interaction. Now the question arises, what is protein-protein interaction? There are various proteins inside the living system that interacts with each other to bring about various changes inside our body. To detect those interactions, this yeast to hybrid system is used. It is extremely cost effective and very sensitive and very accurate process to detect protein-protein interaction. The example of the protein-protein interaction is ligand receptor interaction, second messenger activation, or in case of transcription there are various kind of transcription factors which interact with each other in the transcription process. So these are the example. Here in this diagram it is shown that a protein embedded in the membrane of the cell is interacting with 6 other proteins, and most of the functions in our body is occurring through this kind of interaction. Now, the yeast to hybrid system uses the transcription process to make the prediction about protein-protein interaction. The system requires 2 yeast hybrids be prepared called bait-prey system. The bait protein is fused to a transcription factor DNA binding domain. The other prey protein is fused to transcription factor transcription activation domain. Here it is seen that the Y protein is fused with activation domain, and this fusion is called prey fusion, and X protein is fused with DNA binding domain, and this fusion is called bait fusion. Next, in yeast, GAL4 protein, which activates a transcription of a reporter gene, is used in this process. The GAL4 protein has 2 domains, DNA binding domain and activation domain. These domains are stable when separated, but activation of the RNA polymerase requires the interaction with activation domain, which in turn requires the positioning of the DNA binding domains. Hence the domain must brought together to function properly. So what happens is that GAL4 protein has 2 domains, activation domain and DNA binding domain. Okay, they are perfectly stable when they are separated, but for the activation of the RNA pol, the interaction between activation domain and RNA pol is required, and this activation only occurs when DNA binding domain binds with DNA and also interact with activation domain, and then only activation domain recruits the RNA polymerase and the transcription process begins. Next, genes that encode GAL4 proteins, activation domain and DNA binding domain are manipulated using recombinant DNA technology to produce 2 fusion proteins. We all know that the DNA or the genes are manipulated through recombinant DNA technology. Now the question arises, how these fusion proteins are produced. The 2 domains of the GAL4 protein is separately cloned inside 2 plasmid. One plasmid contains DNA binding domain with X target protein. Here it is shown, the plasmid called bait plasmid containing X target protein, and gal4 binding domain, DNA binding domain. This plasmid also contain a marker, that is tryptophan marker, which is useful for the screening process. Next, what happens? This plasmid is plated on a media that lacks tryptophan. So only the plasmid that took up this tryptophan will survive on that media. So those plasmid are screened by this process. Next, what happens? The other plasmid contain activation domain fused with Y protein. Okay, here the Y protein containing activation domain. This plasmid contains a marker called leucine marker. This plasmid then plated on a media that lacks leucine. So only the plasmid that contain the leucine will survive on that media. So they are also screened in this process. Next, what happens? Next, a yeast strain, that is genetically modified yeast strain lacking leucine and tryptophan, is inoculated in a media lacking leucine and tryptophan with bait and prey plasmid. So what is happening? Next, yeast strain is used that lacks leucine and tryptophan, and they are inoculated with prey plasmid and bait plasmid in a media that lacks tryptophan, leucine and tryptophan. Okay, so in this media only the transformed cell will survive, that is the cells that took up both the plasmid will only survive. So in this way the transformed cell are selected. Now let's talk about what is happening inside the transformed cell. Inside the transformed cell, bait protein and the prey protein is present. Here inside the transformed cell, bait protein containing DNA binding domain, prey protein containing activation domain is present. For the activation of the transcription of the reporter gene, what is needed? First, DNA binding domain of the bait protein must interact with DNA, then it interacts with activation domain. While they are interacting with activation domain, bait and prey interacts with each other, and then activation domain recruits RNA polymerase. Hence the reporter gene is transcribed and we get the transcription product. And if these two proteins are not interacting with each other, the transcription will be off, so we will not get the expression of the reporter gene. Okay, and this way we can say that two proteins are interacting. The expression of the reporter gene, we can observe the expression of the reporter gene through electron microscope. Also, another process, we can observe the expression of the reporter gene. What is that process? In most of the cases, when this prey and bait are interacting with each other, they produces a colored reaction. So when the transcription we get the transcription product, in most of the cases the transcription products are the colored product. So by observing those color, we can say that those two proteins are interacting. This is it, guys. That's all for today. I have talked about yeast-2 hybridization in detail and also talked about protein-protein interaction in briefly. So if you have any question, do comment, and if you like the video, do not forget to hit the like button and share with your friends, and do subscribe to my channel for more video. Thank you.